rotation, but it only takes place in the iliolumbar ligament. So rotation is minimal, but it is present. So it is not completely zero, but it can be nullified. So the answer is rotation. Now to know more about the vertebral column. The spine consists of 24 movable bones. These bones along with the ligament form an S shape. There is cervical spine, thoracic spine, lumbar spine, sacrum and coccyx. The cervical spine, which is C1 to C7, it is the most flexible part and most common area for injuries. The movement here are flexion, extension, lateral flexion and rotation. The next one is the thoracic spine, which is T1 to T12, and 10 of them are attached to the ribs. So it is not a very quite mobile or flexible region. So the movements here are flexion, extension and lateral flexion. The third region is the lumbar region, which is L1 to L5, and we've talked about it a lot. So next is 5 sacrum. There is 5 sacrum and 4 coccyx. So that's it, guys. Thank you for watching. Please do follow Mentoria Medicina and wait for the next question. Please don't forget to like the video.
Medial epicondyle. As you can see, the figure shown on the question is the lower end of the humerus that is connecting the ulnar bone and the radius bone. So what are the joints that is connecting the humerus with ulnar and radius bones? The ulnar bone is connected to the humerus with the help of trochlea, and the name of the joint is humero-ulnar joint. The radius bone is connected to the humerus with the capitulum, which is also known as the head of the radius, and the name of the joint is humero-radial joint. Let's talk about the epicondyles. There are two epicondyles present, the lateral epicondyle and the medial epicondyle. Both of them are extracapsular. So, I am going to give you a brief description about the medial epicondyle. Medial epicondyle is located on the distal end of the humerus, as you can see on the marked area of the figure that is shown on the question, which is inferior to supracondylar ridge and proximal to olecranon fossa. The muscles that is arising from medial epicondyle gives rise to 4 superficial muscles: the flexor carpi ulnaris, the flexor carpi radialis, the flexor digitorum superficialis and the palmaris longus. As you know, the palmaris longus is absent in 10 to 20% of humans. So, when there is an inflammation in the medial epicondyle, it leads to medial epicondylitis or golfer's elbow. It is also similar to tennis elbow. The main causes of golfer's elbow beside golf spot are racket spot, throwing spot, weightlifting, forceful or repetitive occupational movements. The main symptoms shown by such patients are stiffness, weakness, numbness and pain. So the proper prevention of golfer's elbow are taking proper rest, strengthening the forearm muscles, stretching before any activity and lifting the weight properly.
The answer is middle meningeal artery. The point shown in the picture is pterion. Pterion is the thinnest part of the skull where the frontal bone, the parietal bone, the temporal bone and the sphenoid bone meets. The middle meningeal artery goes beneath the point. In this picture, we can see the frontal bone, parietal bone, sphenoid bone and temporal bone. Pterion is the point where all these bones meet. The middle meningeal artery can be seen as ascending just beneath the pterion. In this picture, we can see the fracture of pterion and the rupture of middle meningeal artery causing bleeding into the epidural space, and the bleeding causes hematoma, which is a biconvex shaped hematoma known as epidural hematoma. The surface anatomy of pterion. The pterion lies 4 cm behind the frontozygomatic suture and 4 cm above the center of the zygomatic arch. It is the common point where these two lines meet. In this picture, the two lines are shown as A and B. The length of A and B is same, that is 4 cm. The fracture of the pterion causes rupture of middle meningeal artery and bleeding into the epidural space, causing epidural hematoma.
The neurotransmitter secreted from the arrow marked area is option C, dopamine. The arrow mark shows the substantia nigra part of the brain. This question shows the coronal view of the brain. Substantia nigra is a basal ganglia structure located in midbrain. In Latin, substantia nigra means black substance because its appearance is darker due to high levels of neuromelanin in dopaminergic neurons. Substantia nigra consists of two parts, pars compacta and pars reticulata. Substantia nigra plays important brain functions such as eye movement, motor planning, reward, addiction, learning, etc. Most of dopamine neurons of brain originate in midbrain and are found in substantia nigra and ventral tegmental area, an adjacent area of substantia nigra. In substantia nigra, the dopamine neurons are present in pars compacta, while GABA neurons are present in pars reticulata. Many of dopamine neurons of substantia nigra project to striatum, another part of basal ganglia that is made of caudate and putamen. This pathway is known as nigrostriatal dopamine pathway. Dopamine is both excitatory and inhibitory neurotransmitter. It depends on the receptor it binds with. Dopamine is monoamine neurotransmitter because it is derived from amino acid tyrosine, which is converted to L-dopa, which is decarboxylated to dopamine. It is also a catecholamine because it contains a catechol nucleus. The main functions of dopamine are: it inhibits unnecessary movement, inhibit the release of prolactin, it stimulates the secretion of growth hormone. Due to lack of dopamine in pars compacta, Parkinson's disease occurs. It is a neurodegenerative disease. Its symptoms are tremor, akinesia, bradykinesia, stiffness, fatigue, etc. Now let's discuss the other options. Acetylcholine is mainly excitatory, but it is inhibitory at the parasympathetic endings of vagus nerve. GABA, also known as gamma-aminobutyric acid, is inhibitory. It reduces neuronal excitability. Glutamate is excitatory. It regulates CNS excitability, memory, and learning.
The orthodromic conduction in a neuron is due to the presence of chemical mediators in the presynaptic terminal. Before going into the depth, we can revise some basics of neurons and neuronal conduction. This figure shows the diagram of a neuron. The basic functions of a neuron are to receive signals or information from the other neurons of the sensory receptors, to integrate these incoming signals within the cell body, and to communicate these signals to the target cells or other neurons, muscles or glands. The anatomy of a neuron. The neuron have a cell body which is called the soma. The soma has nucleus and nucleolus within it. It also has neurofibrils and nissl bodies. Various processes or appendages are extending from the cell body. These include many short branching processes known as dendrites and a separate process which is a long one than the other dendrites known as the axon. The dendrites take up the signal or the information from the other neurons and process in the cell body. The cell body, we know it integrates the information from the dendrites and produce an action potential which is conducted via the axon to the other neurons. Now, the axon. The axon arises from the cell body at a specialized area called the axon hillock. Many axons are covered with a special insulating substance called myelin, which is produced from the Schwann cells. The axon splits up into many branches and develops bulbous swellings known as axon terminals or nerve terminals. These axon terminals make connections on target cells. This figure shows the three types of analog signaling found in neurons. The first one is orthodromic signaling, where the somatodendritic voltage changes are propagated to the axon from the soma. That means the signals are from the soma or the cell body to the axon. The next one is antidromic signaling, where the axonal voltage changes are propagated backward to the axon initial segment and the soma. That means from the axon to the cell body. Then the third one is local signaling, where the axon voltage changes are propagated within the axon only. That means these signals are not escaping from the axonal compartment. Now, most of the neurons are orthodromically conducting because the chemical mediators are located in the axon terminals so that it can be released into the synapse and action potential can be initiated in the postsynaptic neuron.